GATB 2020 is knocking at your door and the deadline is approaching. You suddenly wake up and found yourself inside this sea and you don't want to drown. You want to fight back and ace this exam. So this particular playlist is for you. This is called GATB 2020 5 days to deadline. This playlist might be a savior boat for you, which can help you to ace this exam. So keep on watching my channel and don't forget to subscribe and hit that bell icon for notifications. So in this video, we'll solve questions from JNU CEEB last 5 years, and the topic is cell biology. I hope this would be very informative for GATB 2020 preparation. And please watch this video till the end and let's begin. So I have collected questions from 2015 to 2019 and arranged them in a logical framework which would be helpful for your preparation. And by the way guys, I have also qualified JNU CEEB long back in 2016, and in this year I secured a rank of 02, so you can rely on me and I'll try to share all my tips and tricks with you guys which would help you. Now first question is, the alpha subunit of the G protein contains the binding site for cyclic AMP, cyclic GMP, GTP and ATP. So it's very easy, it is GTP, right? So let me revise or refresh your memories on G protein coupled receptor signaling. First, in the sequence, the ligand binds, and ligand is generally proteinaceous in nature in this case. That leads to a conformational change in the G protein, which leads to separation of the G alpha subunit and also replacement of the GDP in that G alpha subunit to GTP. That activates G alpha subunit, which activates adenylate cyclase, and as a result, ATP gets converted to cyclic AMP. And with the rise of cyclic AMP in the cytosol, several kinases such as protein kinase A gets activated, and later they get translocated into the nucleus. The catalytic subunit get translocated into the nucleus and the cellular events takes place. So the point that you need to remember, and the point which is very important, is in a GDP bound state the G protein is inactive, not active. So it's like an off state, it's like a toggle switch. But in a GTP bound state it is active, and the conversion from a GDP bound state to a GTP bound state is done by molecules such as GIF, guanosine nucleotide exchange factors. So in a GTP bound state they are in an on configuration, but they cannot stay on forever, right? So somebody has to switch off this signaling as well, and that is done by GAPs or GTPase activator protein, which stops the signaling. So this angle or this particular concept is very important from an exam point of view because they ask a hell lot of questions. Moreover, many toxins such as cholera toxin performs modification on G alpha subunit such as ADP ribosylation. So ADP ribosylation leads to locking this particular G alpha subunit in a GTP bound state. So the G protein subunit always becomes active regardless of the presence of the ligand. So this constitutive active state is underlying many diseases such as cholera, diphtheria, etc. So this particular point is very important for any MSc exam. Next question says, which of the following is not a signaling receptor? Mannose receptor, toll-like receptor, vitamin D receptor, and thyroid receptor. So the correct answer is mannose receptor. So let me introduce you. So first we know that vitamin D3 receptor is a intracellular receptor. So it stays in the cytoplasm, and whenever the ligand binds, it will translocate into the nucleus and give rise to gene transcription. Whereas the thyroid receptor is already bound to the thyroid response element, and upon binding to the ligand, which is thyroid hormone, it starts transcription activity because it can interact with coactivator and histone acetyltransferase, etc. Now toll-like receptors, we have learned about toll-like receptors in immunology, and they are like pattern recognition receptors. So they identify several components of the bacteria or pathogen, and toll-like receptor has a specific signaling paradigm. Now lastly, we have mannose 6-phosphate receptor. So mannose receptor, these receptors bind to phosphorylated mannose residues, but this is very important in terms of transporting lysosomal hydrolase into the specific lysosomal compartment. But it is not a signaling cascade. So it is a receptor ligand interaction obviously, but it's not a signaling cascade. So the correct option was actually mannose receptor. Okay. Next question says, which cell organelles containing hydrolytic enzymes are called? This is very easy. This would be obviously lysosome. So this is just like a 9-10 standard question. So just to remind you that all the damaged proteins and inefficient proteins actually are uptaken by the lysosome, and lysosome secretes the hydrolase enzymes which degrades them, right? And also there are endosomes which can contain senile organelle, or it can contain pathogens such as virus and bacteria, which can also fuse with the lysosome, and lysosomal content can degrade them. So this is the function of lysosome. Next we talk about cytokine. Cytokine released from cell A stimulates an adjoining cell B. This is an example of autocrine signaling, endocrine signaling, paracrine signaling, and cytocrine signaling. Obviously the answer would be paracrine signaling. Because, so let me tell you what are the schematics of signaling. So first, communication modes that are used by the cell could be diverse. First mode of signaling is endocrine signaling, where the signal is released in the bloodstream and it travels through the bloodstream and can act on a cell which is in a very distant location. For example, think about any hormone, right? For example, thyroid hormone. It is secreted from the thyroid gland, which is in our neck, or it might work on your muscles, which is present far apart. So this mode is endocrine. There could be paracrine, where the cells are quite close to each other, and that mode would be paracrine mode of signaling. And then there is an autocrine mode of signaling, where the cell secretes the signal and responds to its own signal. So it is autocrine mode of signaling. Both paracrine and autocrine mode of signaling are pretty much common in case of immune system, whereas other type of paracrine signaling involves the synaptic transmission. It's also a paracrine signal because the two synapses are actually two cells and they are close to each other, and chemicals are transmitted in between them. Okay, next question says, which of the following is not true for mitochondrial electron transport chain? Complex 5 is ATP synthase as well as an ATPase. Obviously that's true. Now ATP synthase is a proton pump. Let us see whether that is true or not. That is actually true. Cytochrome C is a part of complex 4. FADH2 is fed into the electron transport chain through complex 2. So the correct answer, let us revise this and then we would understand the correct answer. So here you can see the NADH is converted to NAD, or FADH is converted to FAD, and the electron is transported through this electron chain, right? And now what happens is like protons move from the cytoplasm to the intermembrane space of the mitochondria, and there is a proton accumulation in the intermembrane space, which leads to a proton gradient that is utilized by the ATP synthase. And using the proton gradient, proton can come in through this ATP synthase, and that leads to production of ATP from ATP. And point to be noted, cytochrome C is a mobile electron carrier between complex 3 and complex 4. So these are not an integral subunit of complex 4. So that is why the previous answer, option C, would be incorrect. So this is not true, right? So let's move on and try to understand the next question. All of the organelles can be isolated in intact form except which of these following? The correct answer would be endoplasmic reticulum. I'll tell you why endoplasmic reticulum cannot be extracted in an intact form, or it would be very hard to extract it in an intact form. So in cell fractionation technique, you can lyse the cells and try to put it in an ultracentrifugation tube, and in the ultracentrifugation tube what you do, you put it in the ultracentrifugation, and then at several speeds several organelles are fractionated, and from that fraction you isolate them. So you would have different fractions designated for peroxisome, lysosome, nucleus, etc. But for ER, the problem is ER is continuous with the nuclear membrane, or it is continuous with the plasma membrane as well. It's actually a part of the plasma membrane, the membrane system, right? So whenever you try to extract them, they get broken down into small spherosome like vesicles. So that is why it's very hard to isolate ER in an intact state. It's almost impossible. So that helps us to understand the concept. And next we move on to the signal sensor for a two-component signal transduction pathway is histidine kinase, serine kinase, response regulators, repressors. The option is histidine kinase. So histidine kinase and two-component signaling system I have explained in a different video. I'll put it shortly. Now, which of the following is true regarding the extraction of membrane proteins? Integral membrane protein is removed by change in pH. Unlikely, because integral removed proteins are heavily attached in the membrane, deeply embedded. So if you want to really get rid of them, you have to use high concentration of detergent. Peripheral membrane proteins can be removed by urea. It is actually true, because ureas help to remove the hydrophobic interaction with the membrane phospholipids and try to remove it. But it is only true for the peripheral protein because they are loosely attached. If it is an integral membrane protein, it is not possible. So the correct answer is B. So just to let you know that when we add detergent, the detergent micelle actually take away the plasma membrane, and also detergent form a coat around the proteins, and that is how the proteins could be isolated. What function you might assign or postulate for a polypeptide having zinc finger motif? It's a signal transduction protein, transcription factor, growth factor receptor, cytoskeletal element, and the answer is transcription factor, because we know that zinc finger domain is a characteristic DNA binding domain which has zinc coordinated from cysteine and histidine residues, either cysteine or histidine or a combination of both. So they can bind to specific region in the DNA. Okay. Now, for an example, the steroid receptors have zinc finger in their DNA binding domain, so they could be most likely transcription factors. Other than cytoplasm, the intermediate filaments can be found in which of these following organelles? The correct answer here would be nucleus. So let me remind you that inside the nucleus there is a framework of intermediate filament, and that is known as lamin. Lamin is one type of intermediate filament which is residing just inside the nuclear membrane. Many proteins are found completely outside the bilayer on either extracellular side or cytoplasmic side, but are covalently linked to the membrane bilayer. This is achieved by which type of anchor? So here these are actually lipid based anchors. For example, GPI anchored protein. Now GPI anchored protein is actually have a GPI anchor which is a lipid anchor, and the protein is transported onto the anchor. As you can see here, the protein is transported here on the GPI anchor, and this is how the protein turns out to be floating from this GPI anchor, and it's just like a hook, and the way you tie something from the ceiling or something like that. So this is very easy. Next, which of the following is not a signal transduction event? Increase of intracellular calcium, activation of phosphodiesterase, glycosylation of EGFR, binding of GTP to G protein coupled receptor. So all of these things, you know, calcium is a second messenger, so elevation of calcium level has a message for the cell, right? If you want to learn about more in terms of the second messengers, the link is in the I button. So press the I button for all the relevant links. Activation of phosphodiesterase is another important signaling in terms of G protein coupled receptor regulation. Now, binding of GTP to G protein, we have already learned it's an important signal transduction pathway, whereas glycosylation in the EGFR helps in the protein stability and protein delivery. So N-linked glycosylation takes place inside the endoplasmic reticulum, where an N-linked glycosylation tag is transferred on a native peptide on the asparagine residue with the help of oligosaccharyl transferase. Okay, so this is basically a glycosylation reaction, and this glycosylation reaction can take place in the endoplasmic reticulum. So this is not a signaling event. Which of the following is associated with the protein cytoskeleton composed of lamins? So we have already reviewed this question previously. This question came twice in a row. So you can see this kind of concepts are very important. So this answer would be nucleus, because inside the nucleus there is framework of lamine. So it's just like a metal framework inside a tent, and on top of this framework the nuclear envelope is situated. So lamine has characteristic function of intermediate filament, and there are 3 lamines present in eukaryotes or in higher primates. Now in eukaryotes, centromeres are constitutive heterochromatin or facultative heterochromatin, and always present in the center of the chromosome, not required for segregation of chromosome between daughter cells. So they are actually constitutive heterochromatin. That means they can never be converted to a euchromatin state. They are permanently heterochromatin. Now this option, facultative, is wrong. Always present in the center of the chromosome, this cannot be true, because there are many cases like telocentric, acrocentric centromeres, right? So the centromeres are not always in the center of the chromosome the way it is depicted. So just to remind you that the centromere has specific type of histones which is known as CENP-A, and these are actually constitutive heterochromatin, but the kinetochore portion of the centromere is very important because it attached to the microtubule, so it has a role in division rather than gene expression. Okay, so the correct timing and positioning of the cytokinesis in the animal cell depends upon sliding movement of a band of microtubules around the circumference of the cell, contraction of the band of actin around the circumference of the cell, movement of mitotic spindle fibers, endocytosis of the plasma membrane around the equator of the cell. So here is a cell which is about to go in a cytokinesis. So from both sides of the cell there would be a contractile ring which is consisted of actin and myosin. So it just contracts like a muscle and shorten or pinch the membrane space between them, and that is how the 2 cells are separated in cytokinesis process. So it is very important to understand the contraction of actin myosin actomyosin contractile ring is important for cytokinesis. Nissl bodies are rough endoplasmic reticulum involved in protein synthesis in kidney cells, or smooth endoplasmic reticulum involved in metabolism in neurons. Rough endoplasmic reticulum and are involved in protein synthesis in the neuron, part of cytoplasm along with mitochondria that provides energy to the neuron. So the option correct is C, because inside the neuron there are very prominent endoplasmic reticulum, very elaborated network of endoplasmic reticulum which is known as Nissl's body. So Nissl's body, and also people start doing Nissl staining. This is a characteristic example of Nissl staining. What you can see here is all the endoplasmic reticulum in the cell body of the neuron. Now protein glycosylation generally occurs in endoplasmic reticulum only, Golgi only, and endoplasmic reticulum and Golgi both. So let me tell you, endoplasmic reticulum and Golgi both are the place for glycosylation. Generally, N-linked glycosylation is unique to endoplasmic reticulum, but in general, glycosylation also takes place in the Golgi. So remember this picture. In Golgi, ER, both the places glycosylation can take place. Which of the following amino acids are not phosphorylated by kinase? Serine, tyrosine, histidine, tryptophan, and the correct answer here would be tryptophan. So here you have phosphoserine, phosphothreonine, or phosphotyrosine, even phosphohistidine, but you have never heard about phosphotryptophan, right? Because phosphorylation of tryptophan doesn't even occur, okay? And there is no kinase such as tryptophan kinase. Migration of the cancer cell from one site to other site is known as intravasation, extravasation, metastasis, or extravasation. So the correct answer is actually metastasis. So the correct answer here would be on answer C, metastasis. So imagine there is a skin cancer. So here is the primary site for cancer, but eventually it would be shifted, it would be spread all around the body, and this situation is known as metastasis. So one cell which is malignant can move into the bloodstream, and through which it can infect a second organ, and in the second organ it can also lead to cancer. So that is the biggest problem for cancer. You irradiate in one particular situation, but by that time it has already moved to other places. So that's a big problem in case of cancer or chemotherapy. So that is why we need to understand the concept of metastasis, right? So what is the basic difference between cancer cell and the normal cell? Cancer cell divides continuously, but normal cell do not divide continuously. Yes, there are cells in the body which divides quite a lot, such as the stem cells, such as the bone marrow cells, but there are many cells such as skin cells, neuron, they don't at all divide, and they rarely divide. So normal cells are immortal, but cancer cells are mortal, which is totally false. It's just the other way around. Normal cells are bigger than the cancer cells. It's also other way around. And cancer cell can divide, but does not differentiate like normal cells. This is not true. So all of these things is not true. The option A is correct. The most common form of human cancer is actually carcinoma. So carcinoma is the cancer for epithelial tissue, whereas sarcoma is cancer for your myoepithelial cells, connective tissue, etc. Now here, the leukemia is the cancer for the blood cells. So you can clearly understand the most common one is the carcinoma. Cyclins modulate the progression of the cell cycle by activating mostly tyrosine kinase, activating mostly serine threonine kinase, G protein, etc. So the correct answer is serine threonine kinase, because these cyclin and CDK complexes, they are actually phosphorylate the serine residues or threonine residues in the target protein. So they are serine threonine kinases. Okay. Retinoblastoma gene functions in regulating the cell cycle transition from. So the answer wants which phase of the cell cycle is augmented by retinoblastoma gene. So it is actually G1 phase. So let me tell you what happens in the G1 phase. G1 phase is the window in the cell cycle where cell receive external growth factor signal, and the external growth factor signal leads to production of cyclin D, and when cyclin D is produced, cyclin D CDK4/6 complex can phosphorylate PRB. Now PRB generally inhibits growth by sequestering E2F, but when in phosphorylated version it leaves the E2F. As a result, E2F is free and it can transcribe the other cyclins that are required for S phase progression. So that is how the retinoblastoma protein can modulate the events from G1 to S phase transition. So I hope this is clear. Now neoplasia state is best characterized by the cells that divide excessive in number, the cells that are dividing and are cytologically abnormal, the cells show malignant phenotype, the cells differentiate after division. So here you can see a quick overview of neoplasia, dysplasia, these terms. Okay, so this is important. So the cells which are dividing and cytologically abnormal, this is the correct answer. So they are generally abnormal looking cells and bit bigger in term than the normal cells. So this is neoplasia. Then which one is not a pro-apoptotic protein? That means which one of these protein does not lead to apoptosis directly. So let us review the process of apoptosis. So apoptosis can take place by external signal or internal signal. So you can see here the apoptosis is it's undergoing by cleavage of caspase 8, or there could be DNA damage which might lead to pore formation in the mitochondria, and here comes BAX, BAC, bead, all of these proteins actually help to form pore in the mitochondrial membrane, which allows leakage of cytochrome C. Now cytochrome C and APAF1 can ultimately activate caspase 3, which is the executor caspase, and that leads to apoptosis. So obviously we can understand BAX, BAD, bead, these are actually, all of these are pro-apoptotic genes, whereas BCL2, BCLXL, these are anti-apoptotic genes. So this is very important. Last question is, the SRC oncogene is serine threonine kinase, growth factor, non-receptor tyrosine kinase, transcription factor, etc. So don't get tempted by serine threonine kinase. Everything in the world is not serine threonine kinase. So SRC oncogene, which was discovered in context of Rous sarcoma virus, is actually a non-receptor tyrosine kinase. That means it can phosphorylate in the tyrosine residue and forms phosphotyrosine. SRC can interact with several signaling pathway components such as RAS-MAP kinase pathway, beta-catenin pathway, PI3-AKT pathway, and JAK-STAT pathway, and thereby have wide variety of cellular effects. So I hope you enjoyed this video. If you like this video, give it a quick thumbs up, and don't forget to like, share, and subscribe. Do let me know in the comment how do you like my videos. Thank you.